Hello future doctors, welcome to Dipenism. I am Dr. Dipen Shah, and today in this segment we are going to discuss a very important concept of genetics, and that concept is translation. In fact, this is the continuation of central dogma principle given by Crick, wherein in my previous video I discussed about the process of transcription by which the mRNA construction was discussed on the DNA. And in this part, we are going to discuss the second step of protein synthesis, that is known as the translation. So, we are mainly going to discuss the definition and the steps involved in the process of translation. And there are specific two steps, that is the complex formation of amino acid with the tRNA and the polypeptide chain formation. So, let us discuss and define the process of translation. Now, the process by which the sequences of mRNA are read and that is used for synthesis of protein is known as the process of translation. Now the sequences that are present on the mRNA are specifically known as the codons, and the codons code is decoded and they are specifying a particular amino acid, and those amino acids are aligned in proper sequences, and that is how the protein synthesis is done. So that is why this process is known as translation. Now the first step is the process of complex formation. Now as we are aware that the proteins are mainly composed of the monomers, that is amino acids. So the first step in the translation process is activation of this raw materials, and as I mentioned, the raw materials are the amino acids. So first step is activation of amino acids, and the amino acids are going to be transferred by a specific type of RNA, and that RNA is known as the tRNA. So there is formation of a complex which is known as AA tRNA complex. So AA means amino acid and tRNA means the transfer RNA, and there is a bond formation between the amino acid and the tRNA, and that complexes are produced. Now as we are aware, the amino acids are of total 20 types. So there are 20 different types of amino acids which are known as the alpha amino acids or proteinaceous amino acids. For activation of this 20 different types of amino acids, what are required is the energy source. So basically the energy is going to be provided by ATP, and 20 different types of amino acids are activated. But this process requires a presence of an enzyme which is known as amino acyl tRNA synthetase. This enzyme amino acyl tRNA synthetase plays the role in activation of the amino acids, and it will also help in bonding of the amino acid which have been activated with the tRNA. In the initial step, when ATP is forming the in the first step where ATP is actually activating the amino acids, so the amino acid combines with ATP in presence of this enzyme amino acyl tRNA synthetase. It forms amino acyl AMP complex. So basically out of the triphosphates, the AMP part is bonding with the amino acid to activate it, and the phosphate groups are separated out. So first step is amino acyl AMP complex. Now this amino acyl AMP complex will bind with the tRNA, that is the transfer RNA, and the transfer RNA, if we represent by a hairpin model, we are aware that at the 3' end the amino acids can be attached, and as the amino acids are attached at this 3' end of the tRNA, which is known as the acceptor arm, we get the formation of amino acyl tRNA complex, and the AMP is now separated out. Again, this process occurs in presence of the same enzyme amino acyl tRNA synthetase. Some important points to be noted that during the bonding of this amino acid, remember that amino acid has two functional groups. One is the amino group and other is the acidic group. So remember that at the 3' end, the bonding occurs of the COOH group, that is the amino acid part has the acidic part, the carboxylic group is playing the role in bond formation with the tRNA. So this is the amino acid tRNA complex which is formed. So since there are 20 types of amino acids, there will be 20 types of complexes that are formed, which are going to be used in the process of translation and protein synthesis. That means 20 types of enzymes are also available inside the cell, which are going to activate the 20 different types of amino acids. So we are done with the first step, that is activation of amino acids and formation of AA tRNA complexes, which is also known as charging of tRNA or loading of tRNA, or it is also known as amino acylation of tRNA. So we are done with the first step, that is activation. Now we'll start with the second step, that is polypeptide chain formation. So now the next step that we are discussing is the formation of the polypeptide chain. This step mainly involves three sub-steps, that is initiation, elongation and termination, for which there are certain factors which are required in prokaryotes and in eukaryotes. Now in prokaryotes there are 3 initiation, 3 elongation and 3 termination factors which are required. These are all proteinaceous factors, whereas in eukaryotes there are 10 initiation factors, 2 elongation factors and 1 termination factors. The initiation factors are named as IF, that is IF1, IF2 and IF3, whereas for eukaryotes there is a prefix small e in front of IF. So E means eukaryotic initiation factors. Similarly for elongation factors it is going to be EF. For prokaryotes they are named as EFTU, EFTS and EFG. TU stands for thermo-unstable, thermostable and G stands for GTPase activity. Similarly for eukaryotic it is going to be EEF1 and 2. The termination factors, remember that it is not TF but instead it is RF. RF stands for releasing factors that will help in releasing the polypeptide chain. So there is RF1, RF2 and RF3 in prokaryotes, whereas in eukaryotes it is ERF1 because there is only one releasing factor which is involved. So let us begin with the first step, that is initiation in the formation of polypeptide chain. Now for initiation there is requirement of an initiation complex. And this initiation complex is mainly composed of certain components, and one of them is the site for protein synthesis, and that site is known as the ribosomes. So ribosomes are going to be present in the cytoplasm where the entire event of protein synthesis occurs. The other component of the complex is known as the mRNA, which has the sequence of codons, and the third component required is AA1 tRNA. So it is the tRNA having the first amino acid which is required for the process of initiation. Let us discuss each component of the complex. So the ribosomes which are present in prokaryotes and eukaryotes, they are different. In prokaryotes it is 70S type of ribosome, whereas in eukaryotes it is 80S type of ribosomes. The S stands for Swedberg unit, which is the measure of their sedimentation rate. In fact, the 70S ribosome is composed of 30S and 50S components, which are the smaller and the larger subunit of the ribosome. Similarly, the eukaryotic 80S is composed of 40S, which is the smaller subunit, and 60S, which is the larger subunit. Remember that during the initiation step, initially the smaller and larger subunits are separated. They are not attached with each other and they are kept separated by the initiation factor IF3. So it is specifically the initiation factor 3 which keeps them separated, and later on they will be attaching with each other. So if we are describing the prokaryotic protein synthesis, then 30S is going to be the smaller subunit of ribosome. Now the larger subunit is still not attached because of the initiation factor 3. To this 30S ribosome, what is going to come and attach is the mRNA. So remember that the mRNA binds to the smaller subunit of ribosome, and it will have the sequence of codons. And the direction on mRNA, we already know it is from 5' to 3' direction, where 5' end will contain the start codon, and that start codon is known as AUG. AUG codon, it basically codes for amino acid methionine in eukaryotes, whereas in prokaryotes, remember that it codes for N-formyl methionine. So that is the start of the initiation codon. Now the important question is, how does mRNA recognize this smaller subunit of ribosome so that it can come and bind with it? So this mRNA has a presence of a sequence which is consisting of 8 nitrogen bases, and this sequence is present in front of the start codon. 10 to 12 bases before the start codon, there is a sequence of 8 nitrogen bases on the mRNA, which is known as SD sequence. This SD sequence is specifically known as Shine-Delgarno sequence, and this was discovered by obviously these two scientists, Shine and Delgarno, and this SD sequence is going to be present on the mRNA, and similarly a complementary sequence is present on this smaller subunit of ribosome, and that complementary sequence is known as ASD sequence, that is anti-Shine-Delgarno sequence. So this ASD sequence and this SD sequence are complementary, and it helps in binding of the mRNA to the smaller subunit. Remember specifically that in the smaller subunit, it is the presence of 16 srRNA which has this ASD sequence. Okay. Now in eukaryotes, there is this binding of mRNA with the ribosome, which is not performed by SD sequence, but in fact on the smaller subunit, that is specifically on 18 srRNA, there will be a complementary sequence which is complementary to a sequence on mRNA of eukaryotes. Now in eukaryotes, at the 5' end, there is a cap which is made up of 7 Mg, which we call it as 7-methyl guanosine triphosphate. We have studied this during the process of transcription, that at 5' end a 7 Mg cap is attached. So this will have the complementary sequence on the smaller subunit of the ribosome in the eukaryotes, and that will help and the mRNA to recognize the smaller subunit, and binding takes place. So we are done with the discussion of the ribosome. We are done with the discussion of the mRNA in the initiation complex. Now let's come to the A1 tRNA. So once the tRNA comes and binds at the smaller subunit of the ribosome, this loop we call it as the anticodon loop, which has to be complementary to the codons of the mRNA. So if here it is adenine on the mRNA, on the anticodon it is going to be U. If there is U, then it has to be complementary A. G will have complementary C. So if the UAC anticodon is there, then the codon anticodon pairing is perfectly correct, and yes, the tRNA which is coming over here is specifically AA1 tRNA precisely. So at the 3' end, there is A1 tRNA, A1 which is properly attached and which is going to be methionine in case of eukaryotes, and N-formyl methionine in case of prokaryotes. And here this is the 5' end having guanine nucleotide. Now once this attachment is done, this attachment is mainly occurring in presence of GTP. So this attachment is enriched because of the GTP. So remember here now ATP is not involved, instead GTP is the energy source which is involved. The larger subunit of ribosome now comes and attaches with the smaller subunit, and that is done because of the initiation factor 1, and Mg2 plus ion plays a role in attachment of this. Now this larger subunit which is now bonded with the smaller subunit has 3 sites which is known as APE. There is a site, P site and E site. Remember that A site is known as the amino acyl site, where all the different types of tRNAs will come and enter into the ribosome. So that is the A site. P site is the site where it is known as peptidyl site, because here the peptide bond is going to be formed. A site we called it as amino acyl site, because all the different types of tRNAs are going to be entering from this A site. So A2, A3, they are all waiting to enter from the A site. P site, where peptide bond between the amino acids is going to be formed, that is the major role of the P site. Once the tRNA leaves the amino acid, the empty tRNA is going to leave from this E site, and hence this is known as the exit site. So the largest subunit has three sites, A, P and E. Now remember that all the AA tRNA complexes comes and enters from the A site, except the first one, which has directly entered from the P site, because P site was empty. So first tRNA directly comes from the P site itself. So with this we complete the first step, that is of the initiation. Once the initiation is done, now we'll move to the next step, that is of the elongation. Now let us discuss with the next step, that is the step elongation. Now for the elongation process, what happens is the next AA tRNA complex enters from the A site. So this complex is going to be A2 tRNA. Suppose the next codon on mRNA is GGG, then the complementary sequence on the tRNA anticodon loop, it is going to be CCC. So it is the anticodon which is present, then definitely the second amino acid which the tRNA is bringing is for glycine. Now what happens is, once the A2 tRNA is entered and elongation has to proceed, the bond between the A1, that is the first amino acid, and the tRNA is broken. So now as we discussed that the amino acid bonds with the tRNA with the carboxyl group. So that carboxyl group which was bonded with the tRNA, that bond is broken, and now the carboxyl group is free. This carboxyl group of the first amino acid bonds with the second amino acid through the amino group, that is the NH2 group. So remember that the amino acid on the A site, the functional group that is contributing is the amino group. So remember, on the A site it is the amino functional group which is playing the role in bond formation, whereas on the P site it is the carboxylic group which is playing the role in bond formation. And once the bond formation is done, the tRNA, which is now empty, is going to leave from the E site. So the first tRNA now exits because the bond between A1 and A2 is formed. This bond formation occurs using the enzyme peptidyl transferase. So peptidyl transferase enzyme is involved in the bond formation. Also here the energy source is again GTP. So GTP serves as the energy source for the bond formation. Also, once bond formation occurs, what occurs is the ribosome shifts to the next codon, and this is known as translocation. So in every step the ribosome shifts to next codon. So what will happen is, again this A1 A2 tRNA is going to come at the P site because of the shifting of the ribosome. A site is going to be again free, and another AA tRNA complex is going to enter from the A site. This translocation is carried out by the enzyme translocase, for which again the source of energy is going to be provided by GTP. So that is how elongation proceeds. The elongation process proceeds from 5' to the 3' direction. Also remember that during the peptide formation, the peptide bond formation, apart from this enzyme, there is also 23S rRNA playing the role in this bond formation, and hence this 23S rRNA, which is playing the role of an enzyme, it is also known as ribozyme. So ribozyme is the term given to 23S rRNA. With this we are done with the process of elongation. Once the ribosome, by translocating, reaches to the termination site, where it could be having a stop codon either UAA, UAG or UGA, which are the stop or the termination codons which are not coding for any amino acid, then the termination of the polypeptide chain occurs. And once the termination has occurred, the releasing factors RF1, RF2, RF3 and in eukaryotic ERF will help in releasing the polypeptide chain from the last tRNA. The smaller and larger subunits of the ribosome again gets detached and they are separated out. That is how the entire translation process proceeds. Remember that to increase the cellular efficiency, it is also observed that many ribosomes comes and attaches with the mRNA, and this condition we call it as polysomes or polyribosomes, which increases the cellular efficiency by increasing the protein synthesis. So this in detail we have discussed about the process of translation. To revise it, the translation process first required the activation of the amino acids and the amino acids charging with the tRNA. In the second stage, we discussed about the polypeptide chain formation, which involved 3 steps: initiation, elongation and termination. In initiation, we discussed about the initiation complex consisting of the ribosome, the smaller and the larger subunit. We discussed about the mRNA having the start codon, which was the AUG, and we discussed about the A1 tRNA and how it comes and forms the bond. Then in the elongation, we discussed about how the other tRNA complexes are going to enter, how the peptide bond is going to be formed using the specific enzyme and the energy source, and the ribosome shifting to next codon in each step, that is the translocation process. And lastly was the termination, where the stop codons, as soon as they are reached by the ribosome, the process gets terminated. So this we have discussed a very important concept and we have completed the protein synthesis in detail. Stay tuned for my further videos. That's all from Dipenism.